everybody. Thank you for joining us at the COBRE and Opioids and Overdose today at Rhode Island Hospital. We are really excited to be joined by two guest stars with an opportunity to learn about drug checking and through drug trash and the treasures we might find. Today, I have the pleasure of introducing you to two experts, Marya Lieberman, who's a professor in the chemistry and biochemistry department at the University of Notre Dame. And she's interested in the constraints of carrying out chemical analysis and using simple technologies to solve problems outside of the laboratory. So you're going to hear about some of that in her studies with street drugs in the US, but she's also taken this work worldwide to global questions of falsified medications and counterfeit medications, in other words. So these are, we're really lucky to have her today joining us. And also co-presenting is Hirudini Fernando, who is a graduate student in chemistry and biochemistry department as well at Notre Dame. And as a graduate research assistant in Marya's lab, she's been doing a ton of drug checking and data analysis on many of the substances that have been really struggling with in this country, the high potency substances. And so learning about immunoassay test strips is what we're going to dive into today. And we'll hear about some of the award-winning novel processes for drug extraction using test strips that she and the team have been developing. So really excited to hear from Marya and Hiro today, who'll share some of that groundbreaking work on fentanyl test strip extraction with us. We're going to have Q&A throughout. Feel free to use the chat, and we'll look forward to a robust discussion afterwards. So thanks so much, and welcome, Marya and Hiro.
Great. Thank you so much, Tracy, for the opportunity to present our work to this group. And today we're going to talk about using fancy mass spectrometry to learn about the unregulated drug supply. And I want to introduce the actors in this play. I'm Marya Lieberman. I'm an analytical chemist at Notre Dame. And with me is my student, Hirudini Fernando. And there's another player in this presentation, the millions of drug test strips that are currently being used by community health organizations to fight the overdose crisis in the US. So these test strips have a lot of information that they can bring to this effort, and they can help in ways that we're not using them currently. If you want to follow the presentation on your own device, or if you want to see a preprint of Hiru's research results, there are two QR codes here. So just snap a picture with your phone. If you reference the materials or use them in another presentation, just give Hiru and me a shout out for that. One of my activities at the University of Notre Dame is we've started up a lab which we call Notre Dame Serving Community Analytical Needs, or the ND Scan Lab. And a major focus of ND Scan is helping researchers to get started with projects that involve working with high potency opioids. So we help them to get over barriers like regulatory requirements and forming connections to the community. And so, for example, we have lab areas that are DEA licensed and are licensed with our State Board of Pharmacy. And so people can work with controlled substances in those areas. And we also have model documents if other researchers want to replicate these, the permissions and file a DEA researcher license, that kind of thing. I'm particularly grateful for support we've received from the Bioengineering and Life Sciences Initiative at Notre Dame and the Office of Research, which enabled us to set up a suite of instrumentations that we use for chemical evaluation of drug materials. So that includes standard forensic tools like an ATR-IR system, which is used to identify bulk components and cuts in drugs. And GCMS, which is great for untargeted analysis of relatively concentrated solutions. And then as of January, we now have a Shimadzu triple quad, which Hiro will talk about in a minute. And that suite of instruments will be augmented later this month with an LC-QTOF, which is an instrument that lets us do untargeted analysis of trace samples. So I'm really excited about that coming in. ND-Scan and my research group together have a lot of community collaborations. So that includes a lot locally in Indiana, but also nationally. We work with groups like ACDC, the Alliance for Collaborative Drug Checking, which I'm sure many of you are familiar with, and with Tracy's organization like MADS, and also with Remedy Alliance. And so well, what can we do in a lab situation to help in a questions coming from a community perspective? Well, researchers can help to answer lots of different kinds of questions, such as, for a lot of people want to know, can they use their fentanyl test strips to check for fentanyl in their weed? And although there are almost no examples where this adulteration has been verified in a lab, it's a very common hypothetical that comes up on the news and on scary communications to the community. So it's reasonable that people want to know if they can do this. And so because we have access to these substances and we can go into lab and do experiments with measured quantities, we can show that 2 micrograms of fentanyl, which is 1,000 times smaller than the 2 milligram lethal dose, would be detected with a fentanyl test strip. That's good to know. We also work with community health organizations and the manufacturers who make drug test strips and regulatory organizations like AOAC International to make sure that drug test strips work properly for checking drugs. So the test strips originally were developed for detecting residual drugs in urine, and a lot of the early products showed cross-reactivity with drugs like meth or cuts like diphenhydramine. Most of the major manufacturers have now found better antibodies. So there's been a process of giving feedback to the manufacturers, and then the manufacturers have responded to that feedbacks. So that's great because the cross reactions are less of a problem with the more recent test strips. But it's still important to continue to monitor new products coming into the market. And that's because test strips are one of the drug checking methods that are easy to implement in restrictive regulatory environments. And so in a lot of states, there are restrictions on drug checking with instrumentation. And there is also clear access to federal funding for drug checking strips for the test strips. And so millions and millions of these strips are being bought and distributed. And so that means there's a lot of distributors and manufacturers who see a lucrative market and want to jump into it, and not all of them are savvy about the needs for drug checking. Today we're going to focus on some more recent work involving chemical analysis of drug trash, which I'm defining as material that contains drug residues but would normally be thrown away. So things like used syringes, where actually you probably wouldn't throw that away in a regular trash can, you'd put it in a sharps container. But used test strips, empty baggies, butts from smokables, and those materials have tons of information that we can access in the lab. Drug trash has some advantages for drug checking applications, and a major advantage is that in many states, most states now, test strips are not treated as drug paraphernalia. So it makes it easier for community organizations to obtain, store, and ship them to the lab. As we'll see in a minute, the tiny quantities of drugs that are contained in these test strips are well protected. They're stored in porous materials and encapsulated between plastic barriers. All that's needed to preserve the sample for shipping, as we found, is to put the strip back in its foil pouch, which has the little desiccant package that you're not supposed to eat. And then you can place that foil pouch into a small baggie, and that's safe to send in the mail. Sample preparation and analysis, as Hiru will explain, is relatively simple to do. we found that there are a lot of paper processing aids, adhesives, and other background chemicals in the extracts from the test strips, but the tandem MS methodology is really good at screening those things out. So now let's take a closer look at how we can turn drug trash into treasure, starting at the point of use, where a person is preparing a sample for analysis with a drug test strip. And now we're going to switch over and Hiru will present.
Thank you, Dr. L.
Yep.
So most of you might be familiar with this and how people use the test strips. So to run a test strip, a small amount of the substance or a residue from a cooker is dissolved in water. So if the user is testing some powders, normally around 10 milligrams of the substance is dissolved in 5 milliliters of water. So the nominal concentration would be around 2 mg per milliliter. Then after preparing this sample, the test strip is dipped into it for around 15 seconds and then laid flat for around 3 minutes. After 3 minutes, user can read the result. So for this specific example of a fentanyl test strip, a single line shows a positive fentanyl test strip, meaning fentanyl is present in the sample, and 2 lines show a negative fentanyl test strip, meaning there might not be fentanyl in the original sample. But these instructions can vary depending on the manufacturer, the brand of the test strip they use, and specifically the supplies like microscopes that manufacturers provide with the test strips. On this slide, we are breaking down the physical anatomy of a standard test strip. So if you look at the top view of the picture, we can see the user interface of the test strip. On the far left is the sample application area with the wavy blue lines that tells the user which end goes into the liquid and how far to dip. In the center, we have the white reaction area where you can see the results. You'll see the test line and the control line. And in this specific fentanyl test strip, you can see the FIL for the fentanyl test strip and the little hand marks to the place where a person would hold the test strip. If we look at the diagram at the bottom, it shows the 4 overlapping membranes placed on the plastic backing card. It shows the absorption pad or the sample pad, which acts like a sponge to soak up the liquid when a person dips the test strip into the liquid. And then the liquid moves to the conjugate pad, which holds the gold nanoparticles attached to the antibodies. So these antibodies react with the target in the mixture. And then there is the nitrocellulose membrane where the test line and the control lines are present. And then finally, on the far right, there is the wicking pad where all the excess liquid from the sample get absorbed onto this wicking pad. All these printed stickers and plastic backing card kind of encapsulate the sample that is stored in the pad. So when you hold the strip, you won't touch the drugs. Moving on from the physical structure, this slide kind of shows the chemistry happening inside the strip. So these test strips, unlike the very familiar COVID-19 test strip or the pregnancy test strips, these are comparative binding assays. So first we'll briefly look at the setup. The conjugate pad contains the bioreceptor, which is the antibody conjugated to a nanoparticle. And on the test line, there is a competitor. It is the immobilized target on the test line. So For a positive test strip, or when the target is present in the sample, the drug molecules immediately bind onto these gold nanoparticles. So now the gold nanoparticles' hands are full. So when they flow past the test line, they can't grab onto it because they are already holding the drug. So you only see a control line. But when there is no drug in the sample or a negative test strip, the gold particles flow through because there is no drug in the sample to hold onto. So when they reach the test line, they bind to the test line, they grab the competitor and then bind to the test line, and you'll see two visible lines of the test line and the control line. So once the test is done, the strip itself retains a significant recoverable amount of the liquid sample. So we measured the specific volumes absorbed by these test strips with respect to the dipping time. In this plot, you can see the y-axis, that's the average mass of the test strip with respect to the dipping time. We tested until 180 seconds. So as you can see, there is a rapid uptake of the sample in the first 30 seconds, but then the curve flattens out, showing that this saturates. So we measured the specific volumes retained for three different types of test strips: the BTNX fentanyl test strip, xylazine test strip, and the multi-analyte fentanyl and xylazine test strips. For all these test strips, they absorb around 50 to 70 microliters of the sample. This shows that even for different test strips, if you are used properly, it tends to take up nearly comparable volumes of the sample. So this means that a used test strip is like a storage vessel that contains enough sample which we can extract later for lab-based analysis to do more confirmatory testing. So for confirmatory testing in our lab specifically, we use a liquid chromatography tandem mass spectrometry. Like Dr. Lieberman said, we are shifting to a Shimadzu LC-MS-MS now. So tandem MS-MS is like the golden standard in analytical, forensic, and toxicology labs. We use to confirm the identity of the drugs and often to quantify them at very low levels. So if I briefly go through into the technical details on how this tandem MS works, in the LC-MS-MS, the sample is introduced through the liquid chromatography and then ionized. This converts the neutral drug molecules into charged ions in the gas phase. So these ions then enter the first quadrupole, the quadrupole 1, often called the Q1 sometimes. And this quadrupole acts as a mass filter. So basically, it selects one specific ion of interest, known as the precursor ion or the parent ion, corresponding to the specific molecule we are looking for. And everything else gets filtered. Then the selected ion enters the collision cell. In the collision cell, the ion is broken into pieces. These are called product ions or fragment ions. This fragmentation pattern is highly characteristic for a given compound. Then finally, these fragments enter the quadrupole 2. This is again like a mass filter where it is said to look for specific species of the shattered ion. So only if the correct fragments are found, then all the signals hit the detector. So when we are analyzing, we look at the parent ion and all of its fragments specifically to identify what compound we are looking at. So we developed a LC-MS-MS method to analyze 21 unregulated drugs This method included fentanyl, xylazine, carfentanil, and also medetomidine. And our developed method was able to quantify drugs down to a concentration of 0.5 nanograms per milliliter. So this enabled the detection of clinically relevant amounts of drugs like high potency drugs like carfentanil and medetomidine, which are usually found in low mass fractions in a drug sample. So this slide shows how the actual data looks like. So the calibration curve and the extracted ion chromatograms at the bottom are for fentanyl, and these ones are what we did for some standard fentanyl sample. So in forensic and toxicology labs, both the calibration curve and the chromatograms are required to establish the identity and the amount of the sample. So this specific calibration curve was created by injecting the standards of known fentanyl concentration, specifically from 1 to 20 nanograms per milliliter. And we use this linear model to quantify the amounts of unknown fentanyl in samples. So on the bottom, we see the actual peaks. The quantifier or the quant ion is the most intense, and the area under this peak is what we use to calculate the quantities. The qualifiers are the secondary fragment ions that we use to confirm the identity. So combining the cal curve and the linear cal curve and the specific ions, we can confirm the presence of fentanyl and how much there is in a sample. So we have this method, but then our question was, how do we know that this method reliably detect the drug trash? So first we did experiments with pure drug mixture that we made in the lab, and this contained 5,000 nanogram per milliliter of 19 drugs and carfentanil and parafluorofentanyl at lower concentration level. Then we dipped the test strip in this drug mixture and cut the test strip after it dried and then soaked into an extraction solvent. Then we filtered this solvent and analyzed using the LCMS-MS. Then we had few more questions. So is it possible to use different types and brands of test strips? Because, harm reduction organizations give out different types and different brands of test strips for people. So we wanted to check if other brands work with this method. And we wanted to check what is the recovery of analytes over the time. And finally, if we can use this method to reliably test real drug samples. So to answer the first question, whether we can use different types of test strips, we extracted drugs from 3 different types of brands. Like we used a fentanyl test strip that specifically target fentanyl in a sample, a xylazine test strip, and a dual fentanyl xylazine test strip that targets both fentanyl and xylazine in a mixture. The reason for using different types of test strip is that to identify if differences of the antibodies used in these test strips would affect our extraction process. So in the plot, you can see the percentage recovery from each of these types for all the 21 drug analytes we used. The main takeaway was that our results showed that the type of test strip had minimal impact on the extraction process and the LCMS-MS analysis process. Then we wanted to answer our second question, which was the recovery of these analytes over the time. Because generally it takes around 2 to 4 weeks to ship these drug trash from a harm reduction site to our lab. So To simulate that shipping time, we stored the test strips at room temperature for about 4 weeks and then extracted the drugs out of them. As showed in the plot, it shows the recovery percentage for each drug again for all the 4 weeks we analyzed. And we observed that all the analytes were recovered from these test strips even at the 4 week time point, which is very relevant for the logistics of sample shipping. And then, as the title suggests, we wanted to see if we can really quantify samples from the real used test strips. So, and to identify what substance is there and how much it is in there. So the top picture shows the used test strips that we received at the lab that with suspected sample and the test strip result. So in the bottom is the data that we generated from extracting these test strips, and we measured the target analyte and the concentration of each of these targets. For some, you might see, if you look at the first row of the table, you might see that methamphetamine was identified and the concentration was over 1000 nanogram per milliliter, meaning that the concentration of the extract was way higher than the calibration range that we used with the standards. So we show that as a concentration higher than our range. Also, the data shows that users rarely consume a single substance anymore. The supply is heavily mixed or contaminated. And additionally, shows the presence of medetomidine at very low concentrations in a sample like 1 nanogram per milliliter, showing that lab-based drug checking is very important for these highly potent drugs. So one question we faced was, in harm reduction, the reliability of these tools that we use in the field. Specifically, like, do medetomidine test strips provide the accurate results? We received 16 used medetomidine test strips that tested samples or that were originally suspected to contain medetomidine. 7 of these 16 were positive medetomidine test strips, and the rest were negative medetomidine test strips. So we compared the test strip results with our extraction results. In 15 out of the 16 samples we received, which was like 94% of the samples, the MTS results agreed with our extraction results, highlighting the ability of the medetomidine test strips for detecting medetomidine at the point of use. We also had one medetomidine test strip that gave a negative result, but the extracted sample did contain medetomidine. When we measured the concentration with the LC-MS, the concentration was just around 2 nanograms per milliliter. And we estimated the quantity of the original sample using the nominal concentration information we had to be around 600 nanograms per milliliter. When we checked the manufacturer LOD for these medetomidine test strips, it was around 1000 nanograms per milliliter. So what we observed was that this sample was the concentration of medetomidine in the sample was below the manufacturer LOD. So that means the MTS worked correctly, but the limitations are there with their limit of detection.
The value of doing this analysis is both individual, so it's useful information for an individual person, but also general. And that's because each sample really has a story to tell. It's connected to a person's unique experience. And so the analytical findings can help people understand that experience in a new way. And that understanding might help them protect themselves in the future. So for example, here we see we had samples where people reported unusual or unexpected effects, like felt like they were knocked out. And that was a sample that they thought was meth, but really it contained also fentanyl and xylazine. Or a person who went to hospital for what they thought was a ketamine OD, and instead we found quite high concentrations of fentanyl and medetomidine in that sample. Another person said that they slept for days, and that sample contained two different sedatives and a high potency fentanyl analog, carfentanil. So the data that comes from retrospectively analyzing these samples, that can be go back to the people who gave the samples and give them new insights. So the other thing is it lets people see patterns over time. So so for example, we had an array of used syringe plungers from Columbus in 2024, and we saw the rise of the appearance of medetomidine alongside xylazine in these samples. It was also interesting that nearly all of these samples had mixtures of opioids and stimulants in them, although we're not sure whether at what point the mixture was made, whether it was mixed by the dealer or by the user. We believe that these patterns will help to guide treatment and care of people in the community. So in Philadelphia, for example, there was a sudden rise in medetomidine incidence in drug checking samples, and that was mirrored by a rise in emergency room visits that were related to the complications of withdrawal from medetomidine. And then I also want to highlight that in our region, opioids aren't the only concern that we've heard about from our partners in the community. We have a lot of issues with synthetic cannabinoids that are sold as tune or tunchi, which is smokable, and that's associated with overdoses that require emergency medical services. There aren't any drug test strips for these cannabinoids, and the main type of drug trash associated with them is the butts of the smokables. So for these, we're actually focusing on GCMS analysis. Okay, so one more thing I want to pass along is we have support from Remedy Alliance that's giving us an opportunity to do confirmatory testing for anybody who's working with drug test strips and gets an unexpected result. So for example, if the test strip outcome doesn't agree with the user experience, or you think that there might be like a false positive caused by a cutting agent. So these strips can just be packed up and shipped off to me for analysis. The results would be sort of like the series of tests that Hiru talked about for the medetomidine test strips. So if you're interested in this, please let me know, and we can talk about targets and logistics for this. Okay, so I want to just, I think that's the end of the remarks that we prepared, and we're looking forward to getting some questions and talking with you more.
Wonderful. Thank you. Go ahead,
Yeah.
Michaela.
I was going to say it's looking like we have quite a few questions and conversations happening in the chat. So there's certainly a lot of excitement for this topic. And I apologize if I mispronounce anyone's name. Kalyani asks, are the drug samples always dissolvable in water? What are the limitations of drug test strips?
Kalyani, this is a question about the samples for use with a drug test strip, right? Specifically, yes. So in order for the sample to make it to the nitrocellulose part of the test strip, it has to have mobility in water. Other types of solvents don't work on these test strips. So yeah, so solubility can be a problem with some drug materials. So for example, benzodiazepines, have limited solubility in water. Although at this point, we are not seeing false negatives associated with benzos because of the solubility. But yeah, this works for water soluble stuff.
Our next question comes from Chelsea. I apologize, I don't remember which chart you might be referring to, Chelsea, but
Oh, totally.
yeah.
Can I? Hi, Chelsea Shover,
Yeah,
at UCLA. Thanks for your talk.
yeah,
This is super interesting. I just had a
yeah.
moment question with what's figure one in the preprint with the showing the amount that can be extracted over time.
Yep,
And, I'm not a chemist, so this is
yep.
probably an ignorant chemistry question. When you're seeing the amount that can be dis are you using this technique to get quantitative results in the sense that you're able to say, well, this sample has, 14% fentanyl and 2% xylazine, or am I misunderstanding what the quant means here? And then I had a question about that chart.
Yeah, okay. So when we do the measurement, we're always making a quantitative measurement of whatever it is we manage to extract from the test strip. But that does not mean that that's what was in the original drug sample. So one of the things that we found, it's well known that some drugs like heroin are pretty water sensitive. And so once the sample has been made up in water, and if it's sitting around, the amount of heroin will go down, and the amount of 6-acetylmorphine and morphine will go up because those are the degradation products. And so that affects not just heroin, there's several other drugs that have limited stability. We did want to establish that we could reliably recover them from a clinically relevant amount of those drugs would still be present after 4 weeks. Um, so that so that's what that chart is about, is sort of establishing that stability curve. Does that answer your question?
I think so. Yeah, I think it's
Yeah,
basically you're saying that you can recover drug material less and less over time for certain drugs like heroin, which totally makes sense, and that
yeah.
it sounds like it would not be appropriate to use this as a technique to try to quantify the purity or contents of the original drug sample.
So if you want to try to link back to the original drug sample, then you need to have a lot of information about how that sample was prepared. So if the initial sample was prepared with a 2 mg per mil dilution, then I would say that we do have a shot at going back and giving sort of maybe a semi-quantification. So normally for LCMS, we would be able to say, oh, our quant value should be right within maybe plus or minus 20% might
Hmm.
be a reasonable error margin for this kind of method. But I feel like our error bars are going to be a lot bigger for these samples, and that's because the sample preparation is not very well controlled in field settings. But I think like
Thank you.
the result that we got with the metatomiting strips, where Hiro was able to sort of predict back and say it's likely that the reason this strip gave a negative result is because not because the strip failed, but just because there was metatomiting present, but maybe not enough to get over the limit of detection for the test strip.
Thanks so much for that question, Chelsea.
Yeah.
The next question comes from Ken. Is the data being reported back to the original public health agencies for drug presence by area or by region? And then also just a comment that Ken appreciates you rebuking the false narrative about marijuana being adulterated with fentanyl, while
Yeah.
providing practical testing exercises on that topic.
Yes, you have to, I mean, just because people believe things that don't have a lot of scientific basis, that doesn't mean that you can ignore those beliefs, right? You have to take it seriously and Yeah. The data, yeah, so when we're working, for example, with the Columbus samples, we were working with the Franklin County Department of Public Health. And so we would, all that data, we would put it in a spreadsheet that was mirrored to their agency. So they had all that data. And then we had some discussions with them because often it's really hard to for the issue with this data is we can generate tons and tons of data, but what's the actionable data? Like what's the data that will enable the health department to give usable feedback to the people who are providing these samples, right? Those are the people who need access to the data, but it has to be in an understandable way that helps them guide their, I don't want to drown people in analytical chemistry data. So this is, it looks like, Ken, it looks like you have struggled with this.
Yes,
Yeah.
definitely.
Yeah, but if we give the agency the full Monty, like all the data, then they have a choice. Like sometimes the agency will just report presence absence, particularly for high potency substances like carfentanil. Sometimes they'll try and report some relative quantitative information, like this was mostly fentanyl, or the main drug in this was xylazine, or something like that. But
Thank you.
it's always, I feel like we don't have like best practices for how to report this kind of data. And it may depend on who we're reporting to. But when I'm giving the data back to an agency or harm reduction organization that sent me samples, then my instinct is to give them whatever data I found out, I'll give them. So,
Thank you so much.
yeah.
Our next question comes from Carter, and I will do my best to summarize this, but Carter, please feel free to correct me if I summarize. With
Yeah.
various strips being hyper-branded, like with different colors, handles, spoons, do we find it important to clarify the design instead? Just like assuring texts always have the dipstick with the symbol of a hand,
Oh, yeah.
and just
Yeah.
about
Yeah.
communicating
Yeah.
to people.
Yeah. Yeah. This is particularly important with when the sample preparation directions are different, or the strips look really different. Like you go from a blue strip to a yellow strip. That's really confusing to people. I think it really highlights the need to train people in the use of test strips. Don't just give them the strips. You also have to give them a demo is best. If you can demonstrate how to use the strips with equipment that's similar to what they're going to use and a target that's similar to what they're trying to target. So if a person is probably going to be testing like a cooker wash or something, then you have to train them, okay, with cooker wash, you need a small amount of water because the samples tend to be on the dilute side. So you want to use just 1 milliliter of water, about what a person might use to prepare their shot, right? So that kind of information can help people to adapt to changes when you go from one brand to another brand.
Our next question comes from Caitlin. Are there known test stripped cross reactivities with local anesthetics? Kind of a follow-up to that is, are test strips, specifically fentanyl test strips, accurate on crack? They're getting some false positives communicated from their community.
Ooh, okay. So there are many cross reactivities that we see with lidocaine, procaine, and other cane anesthetics. So the answer there is yes, those are known cross reactivities. And this varies like from brand to brand. So different products use different antibodies. And so this is one of the things that the manufacturers are always looking for, better antibodies. And so it's kind of like a part of their secret sauce for their strip. And so that, which is unfortunate because it means that, you may get one strip that works really well and doesn't have these cross reactivities. Another strip, is really sensitive to common cuts in your area. And you can't tell from looking at the package. That's part of the information we're trying to make available through the lot checking project. And so I guess we may touch on that in the lot checking out in the test strip office hours later today. Yeah. And then there's a second question about accuracy on crack. Yeah.
Our test strips, specifically fentanyl test strips, accurate on crack. Participants share a lot of false positives using them for their hard cocaine, and I want to give them empowering suggestions other than just letting them be checked on the FDIR.
Okay, so yeah, false positive. Okay, so lidocaine can cause false positives, and that's a cut in crack, and sorry, not in crack, in cocaine, but I don't know if it's used in crack. Hiro, do you know?
I'm not sure.
What brand of test strips are you seeing this problem with?
Oh, this is so I think right now the program provides our program provides Dance Safe brand fentanyl test
Okay.
strips or
Yep.
BTNX depending.
Yeah.
Um and it was like starting some rumors about, this person put this in my, those kind of things.
Yeah.
And I was, I guess, yeah, looking for ways to expand that without like shutting down their interests, and
Yeah.
like
Yeah. Yeah.
maybe teaching better practices for testing it or something like that. If there was
So
any, I just think of it with like crack not being water soluble. Does that have any effect on it?
I I don't th- I don't think I don't think that that would. I mean, I guess one question is, do you know that there really isn't fentanyl? Like, have you done confirmatory testing?
Yeah, in the one instance, yes, we got our lab
You got a
results
lab
back, and
result.
and it was like phenacetin and cocaine freebase, and that was sort of it.
Okay.
So that was, which to me seems pretty across the board right
Yeah.
now in our
Yeah.
local supply.
I mean, I think Ken's note in the chat is reasonable that the sodium bicarbonate might be having an effect. Um, And so I don't know, but this is a kind of community question. We've done earlier testing. We tested both cocaine and crack formulations with mostly with BTNX strips and didn't see any problems. But the Dance Safe folks that's a WHPM manufactured product, so it might be different. So okay, so we can go down the rabbit hole on this particularly if you can get me the lot number and just like send me by email. I'm going to drop my email in. Please don't sign me up for spam. I'll just drop my email in there. Just if you can look up the lot number on the back of the package, and I'll see if we have some archived, and then we can run it. I have some crack in my safe, so we can run it and see if that, yeah,
I had a question
yeah,
for you all. I was curious, we're starting to see, we always see new substances and emerging on the horizon are more of the orphine class.
yeah,
We lack a test strip to detect this
yeah,
currently. Would this be an approach for substances that we don't yet have a test strip for?
absolutely.
We could
Yes,
test
yes,
on a fentanyl test strip and then send to you to find out if there's an orphine in there.
yes.
Fabulous. Okay.
Yes, as long as that means though, so because the tandem mass spec is a targeted method, that means we need to be looking for that substance. So, for example, morphine, I think is on our list. Is that on the naughty list, Hero?
Not yet, but
Not yet, it's
that's on the
coming.
list. Yeah.
Yes, okay. So right now we can't detect morphine in the sample, because we're looking with a targeted method. Um, the QTOF may be able to detect it, but the sensitivity for the tandem method is 10 to 100 times better. And so for high potency substances, I think the tandem is may give us a best shot at it.
Okay.
Yeah.
Thank you. You have another question, but I think also kind of related to
Yeah.
great question about does conducting these analyses open up your lab to any liability concerns, and question about how to manage that or how you manage that. And kind of relatedly, is this work that could be done in other places? Should it grow? Could it grow to localize this work? I know we're at the edge of newness, so that
Yeah.
doesn't spread very readily, especially laboratory procedures have lots of standards, lots of certifications, lots of things that
Yeah.
happen. On
Yeah.
the other hand, it sure would be great if more labs had the ability to do this, so it could
Yeah.
be more
Yeah.
reliable.
Yeah, the LCMS-MS is a foundational piece of equipment for university mass spec cores. So that means um, any big university could do this kind of analysis. And a lot of tox labs use it. So for hospital labs that are analyzing drug residues in blood serum or urine or something like that. They, uh, I've had, we've had many discussions. So Notre Dame has lawyers, and so we've had discussions about this with the lawyers. And so I think their main concern was that I was going to like start providing evidence for criminal trials or something. And I assured them that that was not my goal. I don't want to do forensic analysis. And so then, yeah, so at that point, there, when we provide data, we're always providing, there's some weasel terms in there saying something like, we're not giving you information straight from God's lips here. This is, this is data that's gathered by human beings, and therefore there's a chance that it could be incorrect. Right. So, yeah. So Sarah, I think your concern is that is certainly a reasonable thing to worry about. So far, we haven't had any issues, and we haven't had any problems also with the DEA in terms of this is part of my research proposal that was approved by DEA. Yeah, Carter is pointing out that shipping samples to CFSRE, which is a DEA lab, that's actually that one of the nice things about that, which I would love to be able to get solid samples that are shipped from community organizations, because when we have a solid sample, we can really do quant analysis to say, okay, the sample is 13% fentanyl and 0.02% medetomidine or whatever it is. And so right now we're sort of inferring the concentration based on this series of, oh, first we made a solution, we ran it on a test strip, and then we diluted it and extracted it. And, that's, it would be better if we could do it direct.
I'm going to jump in with another question just to
Yeah.
sort of broaden. I'm curious how these results in your and the program of testing trash, test strip trash, has kind of landed with policymakers and others kind of beyond. I recognize that many of us are in public health or actually closely in the drug checking community and very excited about these. And obviously academics researching. What's the translation of this in other realms looking like? Has it been?
That's a great question. I mean, I think the issue with the test strip reliability, I mean, that's come up a lot, and I think there's been an effort now to institute some voluntary standards for manufacturers, because FDA doesn't want to deal with these test strips because they're not medical devices. And so that leaves the regulatory authority kind of vague. There isn't anybody who regulates drug test strips at this point. And so having a voluntary standard for manufacturers that's actually a step forward from the current situation. Suzanne, I think you dropped something in as a direct message about, would you mind, because I think that's relevant to the question that Tracy just asked. The thing about the Ohio Board of Pharmacy.
Just appeared in SSP.
Yeah,
But yeah, since we were able to mail some strips and that communication happened with the health department, there was obviously conversations going on with the Board of Pharmacy, who's a little more progressive than some other government organizations around here, but it looked like forward progress and we needed to celebrate that. So thanks, y'all.
yeah, it's great. And there's a note that the Board of Pharmacy in Ohio adopted a rule that decriminalized the drug checking tools, including the test strips. So yeah, if we had some input in that, then that's very satisfying. We have had samples, a lot of samples that we've received in the mail. We haven't had any problems with that. If samples are sent in the mail, putting them in a sturdy envelope is a must. So my favorites so far are these Tyvek mailing envelopes. Those things will, you could roll over them with a steamroller and they will not pop open. It's kind of a pain getting them open when we do get them.
We actually just found USPS's regulatory standards for shipping substances in the mail. So it
Yeah,
breaks down that it has to have an absorbent pad, it has to be in sealable plastic that it can't be contaminated with a liquid, or like you had said, through pressure, like it can't pop open at all and
yeah,
harm somebody that's handling it. That's
yeah.
the main thing. And they're specific about the return address. Like both addresses have to be the same where they're going. So I would assume following those standards for shipping strips just to be safe.
Yeah, I think and really the test strips, they're dry, right? After you use them, you let them sit out until they're dry. You stick them back in the foil pouch. There's like a little packet of desiccant in there, so it keeps them dry. And then you put it in a zip top plastic bag, and now you've got secondary containment. And so that fits that series of requirements. So the actual substances are absorbed on the wicking pad and the sample pad inside the test strip.
And Maria, just to follow up on Chelsea's question have you received international samples in this way of test strips? I was just curious.
I have not. I have not. I have not at this point. There's
Okay.
most of my international collaborators are in Africa, and so there's very little test strip use in those countries at this point. And the drugs are actually they're looking at slightly different drugs. So a lot of tramadol, so much tramadol.
Jody, yes.
This is really such an exciting and overdue area of research that contributes so much to understanding what's going on out there. I'm just wondering about your funding, and also wondering about, I mean, this should, how should this be? Do they need someone at your level running this kind of research, or is this something government public health labs can do, should do?
Well, so CDC has they will take used test strips. That's part of the radar program.
uh,
So they'll take used test strips. I think they don't do LCMS most of the time. They do a direct like a DART analysis with a QTOF. So they're taking the test strip and they're extracting materials directly from the test strip onto, and then analyzing with their QTOF instrument. So there is some activity from the group at CDC.
What should we really be pushing for at the national level, or should this be,
Well,
maybe local health departments should take it
I
over?
think I think the sort of from a public health perspective, then there's sort of a couple angles. One angle is that you're kind of doing surveillance of what's showing up in the drug markets, and that's important because it helps to guide treatment. You can't give a person treatment for a drug if you don't know what drugs they're addicted to,
Yep.
right? So you can't, so it's really important information. And then the information from this kind of drug checking is, again, gives a kind of a new angle when you're talking to a person, like a person who's been using drugs for a long time. They've probably heard it all. And so in terms of saying, hey, here's something that may help this person understand like their risks, from a sort of a new perspective. It just is like a way to open the conversation that might have, with a person who's like heard it all.
Yeah.
Yeah.
Well, thank you so much. This is really
Yeah.
great.
Yeah. Awesome. Good. And Tracy has dropped a link to the NIST publication about their test strip extraction. So that's really, yeah, useful work. Carter, do you have a silly question?
I do. Now, I
Awesome.
know you spent a lot of time working closely with Emmanuel Safiros, developing different projects. He named the Morris reagent after Jeremiah Morris's research.
Yeah.
Is the Lieberman reagent named after you?
No, that's Lieberman with two N's,
Okay.
not me. Yeah, and
Okay.
the thing I love about that is the Morris reagent is that this was a guy who was like I think he was like a master's student, so he got a reagent named after him, which is good. Yeah.
What a way to take us home.
Yeah.
Thank you all again so much for the presentation and everyone who joined us today.